Namaskar. Welcome to our channel Bio by Gulab Sir for Wisdom Coaching Center. As a part of series of lectures, today's topic is law of segregation. As we learn about law of dominance, now second law of segregation, Mendel's second law of segregation. It is already mentioned there, law of segregation. This law is also known as law of purity of gamete. Law of purity of gamete. As you know that in F1 generation, all the plants are heterozygous and hybrid for both factor. In F1 generation, we saw that capital T and small t controlling tall and dwarf together only express only one factor, that is tallness. They remain together. They do not blend. There is no blending of that capital T small t. Same here as the two factors, two characters. Color of the seed is the character, and there are two forms of that character, yellow color seed and green color seed. When plant with yellow color seed cross with plant with green color seed, only yellow color seeds are obtained in F1 generation without blending. That yellow and green do not blend to have something intermediate expression, but gene for yellow color being dominant expressed in F1 generation without blending. But when Mendel has decided to do selfing, he found that these two factors of a character, capital Y and small y, they get segregate or separate during gametogenesis, that is with the process of formation of gamete as a process of meiosis. These two chromosomes, one chromosome having factor capital Y, another chromosome having factor small y, get segregated, separated, and as a part of process of meiosis, that when any diploid cell undergo gametogenesis, meiosis produces gamete with haploid number of chromosome having out of that homologous pair of chromosome, one chromosome in one gamete, another chromosome and another gamete, segregating, separating capital Y and small y through that gamete. So genes are transferred along with chromosome, and therefore this factor gets separated. So Mendel has formulated second law of segregation, that during gametogenesis, this allelic pair of genes, that is contrasting factor, allelic pair of genes gets separated and get into the gamete. And since each gamete has only one allele, not both the allele for a gene, that is known as law of purity of gamete, controlling only one form of that character, either yellow or green, either tall or dwarf. To understand that how chromosomes get separated, here is the diagram. So observe this diagram, what I am drawing. This is a cell with the two chromosomes, yellow and green. So see, I do select yellow color chromosome and green color chromosome, so as to have correct understanding. See, this is a green color chromosome. Now when gametogenesis is there, this is heterozygous condition. For example, this where capital Y and small y. So here this is a capital Y and this is a small y. Therefore, the genotypes are capital Y, small y. But during gametogenesis, that is the process of formation of gametes, as we know that it is a meiosis 1 and 2. During meiosis 1, there is a separation of chromosome. Homologous pair of chromosome gets separated and form two cells. So to understand it, just observe. These two cells are produced with this one. As we know that before gametogenesis, every time these are bivalent chromosome, or these chromosomes having two chromatid, each chromosome has two chromatid. So here you can understand that pair of chromosome that with homologous condition now get separated. So first bivalent chromosome transfer to this cell with this capital Y, and another chromosomes that get separated and transferred to the another cell in the same bivalent condition with small y, small y. Now this is called separation. This one pair of homologous chromosome, one chromosome come to this cell, another chromosome come to this cell during meiosis 1. Now what happened during meiosis 2? Meiosis 2 is mitotic. Being mitotic, there is splitting of chromosome, not movement of chromosome. So see how 4 number of gametes are produced with that segregation and separation of chromosome that you observe. Here the cell with capital Y, another cell with again capital Y. So this is one gamete, this is another gamete, third gamete and fourth gamete. Here this with small y and this with small y. Here what we did observe that homologous pair of chromosome having this factor, one chromosome come to this pole, another come to this pole, and thereby form 2 cells with haploid number of chromosome having capital Y in one cell, small y in another cell. Now see these genotypes, that is what with capital Y and small y, they get separated, capital Y and small y. These were the genotype of this one. Here this cell has only capital Y, this cell has only small y. Thus that 2 allelic factors are segregated, separated, and each gamete has only one allele. These 2 gamete has allele capital Y and these 2 gamete has allele small y. And here the law is proved that during gametogenesis this allelic pair of genes, that is capital Y and small y, get separated, and each gamete has this only one factor, that 50% of the gamete has capital Y and 50% of the gamete has small y. This shows that allelic pair of genes, they get separated and segregated during gametogenesis so as to how each gamete has only one allele. And now because of this only one allele present in one gamete is called law of purity of gamete. That gamete is pure to control that pure trait, either yellow or green. So Mendel proved that allelic factors which remain together in heterozygous condition in F1 generation do not blend, they remain together, do not blend, and they get separated, segregated during gametogenesis. That when F1 generation has selfing, that capital Y, small y get separated into separate gamete so as to represent that gamete with individual factor, that is called law of purity of gamete. And hence Mendel has defined this law as a law of segregation as well as law of purity of gamete. Once again, as a part of definition, that allelic pairs of genes get separated during gametogenesis and assort independently. That what they randomly combine and restore the diploid condition. Once again, allelic pair of genes that present in F1 generation separate during gametogenesis and randomly fuse to restore the diploid condition is known as law of segregation. So as to formulate the law and write it, what is law of segregation is law of segregation. In F1 generation, is a heterozygous condition. That is capital T small t or capital Y small y. This is heterozygous condition. These allelic pair of genes or factors do not blend, as well as they get separated. During gametogenesis. And randomly fuse to restore diploid condition in F2 generation. This is known as this is known as law of segregation and as law of purity of gamete, as each gamete has only one allele to represent a trait factor allele, or a factor has only one factor, or receives has, you can say receive also, or receive only one allele is pure to transfer only one factor to control one character is known as law of purity of gamete. I hope you got this definition also, and law of purity of gamete as well as law of segregation as a concept. Even if you are in doubt, you can ask in comment section. I appeal you to like and subscribe our channel as well as press bell icon so as to receive new videos as and when they are uploaded. Thank you.